Okay, good morning, everyone. I will continue with the genomic selection and helper analysis tools we have on Breedbase. I will focus mostly on the genomic selection tool because many of the decision support tools that are part of the genomic selection, Chris already presented them. So, but you would see them along. Okay, so to just give you an overview of what the genomic prediction process is, you start with a set of individuals, in our case, clones or lines that are genotyped with dense markers, and also that are phenotyped for the traits you are interested. And then you fit a statistical model on this data in such a way that you capture in the best way you can the relationship between the genotypic variation and the phenotypic variation of the trait you are trying to model. Once you have a prediction model, then down the road in your breeding program, all you need is just genotype your progenies, no need for phenotyping, and then you can use the prediction model on the genotype selection individuals to predict their breeding merit or breeding values, which you can use to select parents, do crossing, do evaluation, and again go on and on in the breeding cycle. And what I'm gonna do is how you can use the genomic selection we have for this workflow. And also along the way, again, I'm gonna show you how the input data into the prediction model is through some visual tools and also tabular data forms. And I'm going to show you the process live. I have as a backup slides, but I would try to do it live. So let's do. Can you see the website?
Yes.
Yes.
Okay. So this is an example of a Breedbase site, cassava base, basically, on my local machine. And Chris showed you how you can access the analysis tools. It is this analyze menu, and then you select the genomic selection, and then you would be on the Sol.js start page. And from here, you can start your genomic prediction workflow. And like I said, the first step is to create a training data set, which you can use to train a model. And there are a couple of ways you can start creating a training population. One way is you can use the list approach or dataset approach that Chris today and yesterday, Lucas and Adrian showed you how to create. Or you can directly here search for the trial or trials you want to use in your training dataset. So just for now, I'm going to use one trial to create a training dataset. So let's say I want to use this trial that has the phenotype data I'm interested in, and also for which the clones are genotyped. And then I'm gonna click on it, and then it would query the relevant data for that trial and create a training population for me. Then you would be taken to the training population detail page, where you can see how many, for example, how many clones are in that data set, with how many markers the clones are genotyped, and which genotyping protocol was used to genotype the clones. And then you see the list of traits that were phenotyped in that trial. Below, there are these tools like ANOVA. If you want to see if there is statistical variation in the trait data you are interested, or there is the phenotypic correlation analysis tool, which you can use to see what traits are closely related, what are not, and that you can use to decide on what traits to focus when you do your modeling. There is also the heritability analysis tool here. Chris already showed that in more detail. So let's say you want to train a model or models. And so what you need to do is you just select the trait you are interested. You can do one trait at a time or multiple traits at a time. The important thing to know is that this is univariate modeling, which is just you create a model per trait. But by selecting multiple traits at once, you run the modeling at once for all the traits you are interested. So let's say you select these two traits. So it did the modeling for the two traits, and then you are in the models output page. In reality, this is a long process. So when you request for modeling, you would be asked to submit the modeling job, and then the analysis would run on the servers, and then you wait for an email with a link to this output page, and then you can follow the link to get here. So I did modeling for two traits. Here they are, and this is just a glimpse of the analysis output, like the modeling accuracy for each models and SNP heritability. If you want to see the detail of each model, you can click the model of your interest, and then you get to the specific model detail page. And here on the top is some descriptive detail, like how many lines were used in that model, how many markers, etc. Then if you want to check what kind of phenotype data went into the training model, you can look at the phenotype data in scatter plot. In a scatter plot, you can point at any data point and see which clone that is. And this is good if you have an outlier or outliers. You can see the same thing in histogram format. So you see clearly how the data is distributed. You can look at the phenotype data statistical summary, the minimum, maximum values, mean, median, standard deviation, etc. Then if you are interested to look at if there are genetic, if there is genetic structure in the population, you can do PCA. For example, you can do this using genotype data, and then you get the PCA output like this. This is the two principal components, and you can judge from here if there is any population structure in the training data set, which is important to decide whether to split the training data set into the relevant clusters or not, etc. You can do also clustering if you want. I'm not going to show that here. Then if you are satisfied with the data that went into the model, then you can proceed to look at the model outputs, for example, the variance components. You can look at, of course, to decide if you want to use this model or not, you need to look at the accuracy of the model. And here is the output from 10 false cross-validation tests. You can see here 10 false were used, replicated 2 times, and these are the minimum and maximum accuracy values from this tests, and this is the average value.
Isaac, I have a quick question here.
Yes.
The model accuracy seems to be very low. If that is the case, then is there a way we can improve the model, or if that is the case, how can we do that at this time?
Yeah, so if you are not happy with this model, yeah, it means going back to the start point, creating a new training data set, right? But hopefully somewhere here, maybe you identified why the model might be not good. So let's say if you notice here, there is obvious substructuring of the population, and maybe you want to split, I don't know, maybe you want to split the training population into the number of subgroups you notice here. You can do clustering. For example, let's say you want to split this population into 3. So you can do this clustering approach. Okay, this might take time. But yeah, so you would get the clusters, and then you would know which clones are in each cluster, and then you can decide to use a subcluster to start again to create a training population. The other way is maybe you want to combine this training data set with data from other trials. So in that case, you can just combine any number of trials and do again the modeling until you get to the model accuracy you wish for.
Sure.
Yeah. So for example, if you notice here that there are three subgroups, you can use the clustering approach, partition the data into 3, and then you have here clearly the clusters, and you can download the clusters. Yeah, so from here you can, it is not integrated with the database, but you can take a list of, let's say, this class group 1, create a list of them, and then when you create a list of them, then you can use the list approach to create a training population.
Okay. Sounds good. Thank you so much.
Yeah. Okay, so let's assume you are happy with the model, and then yeah, you want to look at the breeding values uh, of the clones from the model. You can see it in a histogram format. You get a view of the top best performing clones, and if you want, you can also download all the breeding value of all the clones. You can also regress the breeding values to the phenotypic data and then see how it is relating. Then next is, let's say you have this model you are happy with, and you made your parental selections, you did crossing, and then you have progenies from the crossing, and you genotype the progenies, and you put already that data in the database. Now you want to use this model to predict the breeding values of your progenies. So all you have to do is come here, come to this stage, and then you can search for the set of progenies, depending how you are organizing them. If they are just a trial, you can search for the name of the trial that contains the progenies, or if you have a list of them, you can just find the list of the progenies here, and then let's see, for example here, and then you say predict, then you are applying the model on this set of progenies, and then you would get their breeding values. So for example, let's say I applied the model on this set of individuals, then the breeding value of these individuals will be displayed for you in this format, and you can download the breeding values also. If you want to evaluate if there is any improvement in the performance between the set of progenies and the training population, you can use this genetic gain visualization tool, and it would display for you the breeding values of the two populations in this box plot format, and yeah, and then if you want to download the box plot, you can download it, or the data of the two populations. Yeah. Does it make sense, or do you have any questions?
Yeah, makes sense.
Okay.
We also can, I mean, is there a place where we can see the relationship?
The relationship between
Marker and the phenotype?
So this is a selection population, so it has only a genotype data. So but if it was uh, for the training population, you can see it. You can see it like I did here. Oh, but one thing also I just wanted to show you. So one issue when you are applying a model into a selection population is how closely related the selection population is with the training population. So you can evaluate that using the PCA. If you use the genotype data of the two populations, you can okay, a legend is missing here for some reason, but it shows you um, um, if there is any subgrouping between the training population and the selection population, and both populations would be in different colors, so you would be able to tell if there is subgrouping. In this case, you can see that there is no real difference. There is no real grouping between the orange ones and the blue ones, which means they are fairly
Close.
yeah.
Okay.
So you can be sure, yeah, this model is good to use for this selection population. So let me go back. So this, yeah. So do this. So what I just showed is just one model for one trait. But yeah, so when you do prediction for two traits, there are many tools that can help you uh, in making decisions. For example, here, if you want to analyze the genetic correlation between the two traits using the GBVs, you can do that. You select the population for which you want to analyze the genetic correlation for. This process takes a little time. Okay, yeah, it is done. So you can see the genetic correlation between the two traits. You can point at the pair, and then you see what the correlation value is. You can check again if you applied this model on a selection population and you want to check the genetic gain for the two traits at the same time. You can do that too. And you see it in box plots. When you do your selection, if you want to use selection index, you can calculate selection index. This is the form. Let's say you want to give these weights for the two traits. So it is calculating the selection index and also the correlation between the selection index and the GBVs of the traits. It takes a little time. Once you have that, then you would get a rank of the clones based on the selection index. So you see, and you can download it. And this is the correlation between, for example, the selection index, and yes, the selection index and the traits. For example, the selection index and the DMCP or dry matter, there is a correlation of 0.78, etc. So you can use that to judge if you can rely on the selection index. So let's say now you have the ranking of the clones based on the selection index, and you want to select 15% of the clones based on the selection index, and then let's say you want to divide the 15% selected clones into four groups or any number of groups that you want to maybe you want to create some crossing groups. based on the genotype similarity. So if you want that, you can select the selection index data, and then the method now is only k-means. You want to cluster them based on genotype, and then you indicate here the percentage of the population you want to select, 15%, and then you indicate here into how many groups you want to partition them. Let's say into 4. Yeah, so you have the 15% of the population clustered into 4 groups that you can use, for example, as crossing blocks or to design your crossing. And you can download the clusters from here, or everything in any way. Yeah, I think so. I think that's what I wanted to present regarding the genomic selection tool. If do you have any questions?
Yeah, I just have another question here. So is the model only one model? Is it a regression model or are there multiple models?
So we implement a GBLAP method based on a ridge regression BLAP R package. So that's the only method we have now. So do you have like other modeling approach you would be interested?
No, I just wanted. No, I have other. I don't have any other models as such in mind. But I just wanted. Ridge regression, of course, is the most common model used.
Yeah. So yeah, we are applying what is a common approach, but if there is a need for other modeling approach, it is possible to add that option. So you can select whatever model you want to use. Yeah. Okay. So
Yes,
I think
thank you.
yeah. I would continue. Is Adrian here?
No, Adrian left for another meeting.
Okay, so I would just briefly talk the Soljuas tool Adrian is developing. Okay. So we have another analysis tool for GWAS, and Adrian is working on it. And I just want to give you a brief overview how it works. You start with a data set. You create the data set the way Chris was showing earlier, and then you select the trait you want to fit a GWAS model, and then first you can evaluate, of course, the phenotype data in a histogram. Then you have an option to fit the kinship matrix or principal components in the model. Depending on your choice, you just press a button, run SolGWAS, and it gives you the output in such format. For example, the marker effects in a Manhattan plot. And if you want to evaluate the model, you can also look at the QQ plot. And I believe the outputs are downloadable. And I think that's it. We also have a mixed models tool that is under development, should be out very soon. This model also works the same way as the GWAS. Or if you have a data in organize it in a data set, you can select the trait you want to fit a mixed model, and then it has this option to fit any of the factors as you would like, as a fixed factor, as a random factor, and then just easy go. You click go, and then the mixed model would be fit, and then you get the genotype effects, and which you can download or save in the database for later analysis or use. And this is yeah, Lucas is working on this. Maybe Lucas, do you want to add anything more?
No, I couldn't say it any better than you just did. Yeah, that was great. Yeah.
Okay, thank you. Yeah, so yeah. Okay, so yeah, that's what I have. And if you have any questions, please ask. Any questions? Okay, so I would transfer.
Thank you so much, Isa.